Good afternoon, everyone, and welcome to the Society of Wetland Scientists July 2020 webinar. Today's webinar is Can Biochar Increase Carbon Sequestration in Wetland Restoration Projects? And it will be presented by Priyanka Sarkar and Beth Watson. My name is Bianca Wenzel, and I am pleased to moderate this webinar and engage you in a remote learning experience with SWS. For those of you who may not be as familiar with our SWS webinar series, all of our monthly webinars are complimentary to SWS members and are usually held on the third Thursday of the month. Once each quarter, we open our webinars and offer them free to non-members and the public. As you can see on this listing of our upcoming webinars, we have both our regular SWS webinar series in English and also have quarterly webinars offered in Spanish. Our next free quarterly webinar will be held in September 2020, titled Urban Wetlands and Water Management, and will be presented by Bill Dooley. We are proud to announce our SWS webinar series sponsors: the Water Resources, Hydrology, and Hydraulics Education Group, which is a nonprofit group that shares information on water resources, hydrologic and hydraulic engineering to help educate and build a better tomorrow. Please visit, like, and follow this sponsor. And the Whittington Group, which is a natural resource consulting firm that balances regulatory compliance with sound ecological management. You can find their information at the websites listed above. Also, in situ monitoring equipment and software works together to make it easier and more cost-effective to collect, access, and manage water levels, water quality, water flow, and more. And the Wild Note is a field data collection app for environmental professionals that streamlines the process of collecting, managing, and reporting environmental data so you can work smarter, not harder. You can find their information at the websites listed. Please visit, like, and follow our SWS webinar sponsors. We are proud to announce that this webinar may be approved by the SWS Professional Certification Program as applicable for continuing education credits. These credits can be applied to your professional wetland scientist certification or renewal or other certifications. Participation certificates are available through an automated process. You will receive an email from our SWS Managing Director, Michelle Chosick, about 1 day after the webinar. Check your spam email if you don't see it. These certificates are free to SWS members and are available for non-members for a nominal cost of $20. Participation certificates are also available for those who watch webinar recordings, which can be found on our past webinars page or our YouTube channel, complete with multilingual captions. Before we get started, I just want to go over a few housekeeping items. The general format for today will be a 45-minute presentation by our speakers, followed by approximately 15 minutes for questions. All attendees will be muted. For the Q&A, any questions will be asked in the Q&A pane, not in the chat panel. So make sure you do that in the Q&A pane. At any time during the presentation, you can type your questions into that pane, and then participants can upvote particular questions that they want to be answered earlier. The chat pane shouldn't be used for questions, but if you have any technical difficulties or just some general comments you want to write, you can feel free to do that in the chat. As for handouts, a PDF of the webinar will be hosted on the SWS website, but you'll also be sent an email immediately following the webinar, and that will include a survey about your experience. Also, don't forget to tweet about today's webinar at #SWSwebinar. So with the logistics out of the way, I would love to introduce our speakers, and that's Priyanka Sarkar and Elizabeth Watson. This webinar is very special today because it features work conducted through our SWS Wetland Ambassadors program. Priyanka Sarkar is our 2019 Wetland Ambassador, and Elizabeth Watson was her host mentor during this experience. Priyanka is currently finishing her PhD on conservation of a floodplain wetland from the ecosystem services perspective at Assam University. In 2019, Priyanka received the Green Talents Award hosted by the German Federal Ministry of Education and Research. She's also been appointed as a graduate research associate at Drexel University, and she serves as the Asia lead at the Youth Engaged in Wetlands, an international network to enable and empower young people to help support the mission of the Ramsar Convention on Wetlands. Priyanka is very passionate about conservation and sustainable management of wetland ecosystems, as I'm sure you'll see from her presentation. Dr. Elizabeth Watson is an environmental science faculty member at Drexel University in Philadelphia and the wetland section leader at the Patrick Center at the Academy of Natural Sciences. Watson received a PhD in geography from the University of California, Berkeley, and conducted postdoctoral research with the hydrology group at UC Davis and at CICESE in Ensenada, Baja California. Prior to coming to the Academy in Drexel, she worked for the U.S. Environmental Protection Agency in Rhode Island. She has published more than 50 scientific papers and sees herself as part of the paradigm shift that is occurring in coastal management as climate change adaptation and mitigation are largely displacing more conventional coastal management goals. Those are our speakers. You can get more information on them from their bios. But with that, I will hand over control to Elizabeth. Welcome, Beth. We're excited to hear what you guys have to say.
Thank you very much, Priyanka. Okay, I'm having some trouble controlling the screen, Jordan. It says that I'm in control. Okay, I'm able to move forward now. I apologize. Okay, so I wanted to start out by giving a broad overview of how our lab and the students I work with got involved with doing research on wetland restoration and biochar. And this comes back to a restoration project that we've been involved with in California. It's at the Elkhorn Slough National Estuarine Research Reserve, which is in central Monterey Bay. And if you have never been there or heard of this place, it is a very beautiful and interesting and lovely place to work and visit. And I would recommend checking it out in person or virtually as you have time. So one of the main challenges to conservation at Elkhorn Slough in terms of wetland conservation is a history of diking and draining coastal marshes. So in general, on the West Coast, you tend to see marsh vegetation that grows between about the level of mean high water and mean higher high water. And so over the 1800s and the 1900s, there were a considerable number of dikes and levees that were constructed, both as part of the construction of a railroad that bisects the whole estuary, as well as just generally constructing levees to convert the lands for agricultural uses. So for growing crops or for the most part, actually for dairying, for pasture lands for cows. And so what happened over time is that the wetlands, things that had been coastal tidal wetlands, they subsided. So they dewatered, the soil combusted a little bit, they consolidated. And so these places are too low in elevation today to support coastal marsh. And so what's happened is that over time, the levees have breached, or in some places they have been deliberately removed or deliberately breached. But in almost all cases, the marshes are, or what had been marshes, are too low to actually support plants. So what you end up with is kind of a bare mud flat that isn't necessarily functioning very well as the mud flat either. So it's kind of a degraded marsh mud flat mix. And so this is a oblique aerial photograph that shows kind of what this looks like at the landscape scale. So this is a place that had been tidal marsh, 100, 200 years ago. And so what you're looking at here is coastal grassland, some shrubs, some trees, and then this broad area that is mostly mudflat. You can see in green that there's some algae on it. So there's a lot of blooms of kind of opportunistic and nuisance macroalgae. And then for the most part, the vegetation or the marsh vegetation is limited to this very narrow sort of bathtub ring that surrounds what, you know, had been a fairly expansive area of tidal marsh. So why aren't these plants kind of recolonizing on their own? So it's basically it's too low in elevation. And then there have been a lot of other modifications to this environment and this estuary that really decrease the sediment supply. So this is not a place where the wetlands are able to kind of accumulate sediment and build up and restore themselves because of kind of other things that have gone on. And then a second issue is that this is a place that lacks the West Coast ecosystem sort of coastal marsh pioneer, Spartina foliosa. And so there's not sort of that plant that kind of typically might grow on sort of the marsh mudflat boundary and help to kind of trap sediment and jumpstart that process. So there's some kind of anthropogenic and then more natural reasons why it's kind of hard to restore wetlands here. And so in order to restore large areas of tidal marsh at Elkhorn Slough, there's basically a need to bring in sediment. And so what I'm showing you here is a movie that, or a video that shows both the process of moving sediment around and then kind of a landscape scale to get you an idea of the scale of the restoration project that we've been involved with. And so this is a place where they were very fortunate to have a local supply of sediment. And this was generated through a project that occurred nearby. So there's a local river, it's called the Pajaro River. And over the last 20 years, there have been a lot of kind of large flood events along that river that really choked the channel and the floodplains with sediment. And so what it meant was that every time there was a small kind of rainfall event, you flooded both the local community and agricultural land. And so there was a lot of interest in removing the sediment from the floodplain in order to allow the river to, you know, flow to the ocean and not kind of flood the surrounding towns. And so there was basically a source of sediment that could be used to kind of build the marsh elevation up again to restore tidal wetlands that was free, aside from the cost of sort of permitting and storage. And so I believe that the sediment was actually brought to this marsh restoration area within the cost of the flood and floodplain project. So it was something that was not a huge cost to the people who are actually managing the wetland restoration. And so I've been involved with this project in terms of helping to understand patterns of recolonization and also carbon sequestration, which is one of the goals of that project. So at the same time, there was a land management effort at that same place that was kind of following a similar track that involved the removal of non-native eucalyptus trees, which is what I'm showing a picture of here. And removing eucalyptus trees is something that can be somewhat controversial in California, not always, because they can provide important ecosystem functions and habitats as well. However, the land managers at this marine reserve really spent a lot of time carefully documenting whether the eucalyptus trees were expanding, the extent to which they were expanding, and then also their role in providing important habitat. So they compared sort of the insects and the birds and, you know, all sorts of organismal use in groves of eucalyptus trees and compared that to native grasslands, woodlands, and shrublands in the area to try to decide whether it was going to be useful or relevant to take those trees out. And so after the end of this kind of long process of study and consideration, they decided that actually they weren't providing any kind of crucial habitat, and they were expanding dramatically enough that it was worthwhile to take the trees down. And so one of the things that is a consideration in most projects in California is the greenhouse gas footprint of what you're doing. And so there was this idea that the wood from the trees that were being removed could be burned to create biochar, which is could then be used as a soil additive in the restoration project. So this would contribute to sequestering carbon in the restoration project, and it also potentially might improve soil conditions and encourage plant recolonization. So what is biochar? So biochar is a carbonaceous soil amendment that's produced through using an organic feedstock. Most commercially available biochars are produced using some kind of waste product from either agriculture or forestry industries. And so you might have typical types of feedstocks might include things like rice husks or corn husks or you know, products from the lumber industry. And then that material is combusted anoxically, which is called pyrolysis. And so that treatment then produces this basically charcoal-like material. And so this material can encourage plant growth and restoration. And it has a large surface area and very porous structures, which can provide microhabitats for microbes. And then because of that, really encourage a very healthy soil ecology, as well as contributing to carbon sequestration, because it is thought that the biochar is very stable in the soil. However, most of the studies that have focused on biochar have found that there are so many variables, it's hard to make strong generalizations about what the effects are of biochar in soils. So for example, studies that have looked at biochar have tended to find that it can increase plant biomass, it can help with pH balance, it can help with denitrification, promoting denitrification. So that's definitely an environmental issue in this estuary that I haven't discussed, and I'm probably not going to discuss, but you can imagine that this is an estuary in California that's surrounded by agricultural areas. And so, and in some cases, the agricultural is even like multiple cropping per year, so double cropping or triple cropping. And so there's huge issues with water quality because of that. And so the biochar can potentially, you know, provide the structure and encourage the growth of microbial communities that contribute to denitrification. And again, a major concern in the context of all environmental management projects is carbon sequestration, as well as the greenhouse gas footprint of the project. So one of the things that is just really obvious when you start reading the literature or looking into analyses of biochar is that the effects that it have really vary depending on all sorts of different things. So it could include things like the feedstock material, the pyrolysis temperature, how long it's held at that temperature, as well as all sorts of different things related to soil conditions, the composition of the soil, the grain size, the mineralogy, how much organic matter is, the temperature, the moisture, etc. And so this is a really understanding whether biochar can help with, you know, a particular restoration goal is a pretty complicated question. So that's kind of the context on how our lab kind of got interested in understanding the interactions between biochar and restoration. And we have been involved with a number of field studies that really look at the role of, or the potential of this eucalyptus biochar to promote revegetation, as well as things like denitrification. And then we also, a couple summers ago, we did a lab incubation experiment, which we actually grew plants in tidal mesocosms with and without biochar to try to get a little bit more of a detailed understanding on how the biochar, you know, influences soil conditions in the context of coastal wetlands restoration. So the opportunity here for Priyanka was that we had mainly studied these softwood biochars, so biochar that was produced commercially as a forestry product, as well as biochar that was produced from these eucalyptus trees. And so we were interested in understanding kind of a broader range of feedstocks. And so one of the research objectives of the project that Priyanka is going to describe is, first of all, to determine the short-term stability of carbon that's in biochar in coastal wetland soils, really focusing on different kinds of feedstock materials. And then secondly, another issue that often occurs in projects where you put sediments on top of marshes to build their elevation is that the sediments, not in this project in California, but in general, the sediments are from subtidal conditions. And so they can be very organic and sulfidic because of the reducing conditions found at subtidal areas. And so when you go and you put them on your tidal marsh, you're introducing them to an environment with a lot more oxygen. And so then what happens is those sulfides can oxidize to form sulfuric acid. And so and so this is something that can be very detrimental for plant reestablishment. So if you have soil conditions that are like pH of 3 or pH of 4, then those conditions can really inhibit the reestablishment of plants. So those were the things that we're interested in investigating in this project that Priyanka led, really understanding the short-term stability of carbon that's in biochar. And then secondly, thinking about how it might interact with pH. And so with that, I'd like to turn it over to Priyanka, who will describe the study and her results.
Thank you, Beth. Hello, everyone. So okay, so as Beth mentioned, we conducted for our study a factorial experiment during the year of June, during the year 2019, from June to September at the Drexel University campus. And we collected our samples from the Barnegat Bay areas in the New Jersey. This is the study area map showing the sampling stations from where we collected our samples. And this is the the coastal marsh areas. So we collected the sediment samples from those areas. Along with that, we also collected sand samples and also the bay from the bay water from the Barnegat Bay. For our study, we use 5 different feedstocks, namely bamboo, mixed hardwood, mixed softwood, rice husk, and algal biochar, which were all purchased from online store. For the study, we wanted to mix a ratio of the soil and biochar using volume by volume. So we wanted to have a uniform texture for all the biochar types. So for keeping the texture uniform, we grounded the biochar into using an industrial grinder, and we stored it in airtight plastic packets. In the greenhouse, we first started with preparing our soil. We prepared a soil mixture with a ratio of 1:1 of marsh mud and the sand, and those were mixed with different levels of doses of biochar, such as 0%, 10%, and 20%. And the different types, as I mentioned before, the softwood, hardwood, bamboo, rice husk, and algal biochar using cement mixture. The mixture were then finally put into 55 different tree pots, giving a sample number of 5 for each treatment with a total experimental unit of 55. For setting of the experiment, each pot that were filled with the biochar sediment mixture was then transferred into individual food grade plastic buckets with a bed riser placed within it. The pots were incubated in the greenhouse, maintaining a low tide water level, that is 7 inches, using the bay water collected. During the entire study, we maintained the salinity of the bay water as 20 ppt. The incubated pots in each bucket were watered thrice a week with 100 ml of distilled water. In the incubated pot, we inserted a pore water sampler known as the rhizome samplers to collect the pore water at the regular intervals that I will be discussing in the next slides. And the buckets were placed in the buckets with the incubated pots were placed in the greenhouse randomly. This is the slide showing the incubation setup, where the individual buckets with the incubated pots were placed into 4 different tables randomly by generating random numbers assigned against each treatments. This is a different abbreviation for the low dose and the high dose of biochar for different biochar types. For the data collection, we studied the carbon mineralization from the incubated pots over a period of 12 weeks, and by measuring the carbon dioxide and methane fluxes using a portable gas analyzer. The gas analyzer was connected to a closed system gas flux chamber and were covered by a dark cloth for providing a dark condition. Every time before taking gas measurements, temperature in each pot were measured using the temperature probe. For collection of the pore water from the incubated pots, the rhizome samplers, which were put into the incubated pots around 12 centimeter depth, at the 2nd, 4th, 8th, and the 12th week of incubation, some syringes were attached to the rhizome samplers, and pore water were collected into small vials, which were immediately transferred to a cooler box and brought to the laboratory for further analysis. For the pore water, we mainly studied the pH, salinity, and total acidity, whereas the pH and the salinity were analyzed for 2nd, 4th, 8th, and the 12th week of incubation, and the total acidity was analyzed for 3 months, that is week 4, 8, and 12th. For analyzing the pore water samples, the pH meter was used, and for analyzing the pore water salinity, the YSI conductivity and salinity meter was used. To estimate the total acidity of the pore water, the titration method was followed using referring the AFA 2012. Besides, some pore water samples were sent to the US EPA for dissolved organic carbon analysis. For data visualization, the MX Excel 2016 was used, and for statistical analysis such as ANOVA, Tukey post hoc test, and independent t-test, the SPSS version 20 was used. Now results and discussion. This graph showing the carbon dioxide fluxes from the incubated pots over during the study period, where the x-axis showing the weeks of incubation and the y-axis showing the carbon dioxide flux in mu moles per meter square per second. The colored line showing the values of carbon dioxide emission from the treatments with different biochar types such as softwood, hardwood, bamboo, rice husk, and algal biochar. The upper graph represents the low dose incubated pots, and the lower graph representing the high dose incubated pots. The graph suggests a temporal variation in the carbon dioxide efflux for each biochar types, and the value also varied between the low dose and the high dose treatment of the same biochar types. Overall, the data suggests highest carbon dioxide effluxes relative to control value in the algal biochar as compared to other biochar types. So this could be mainly due to the reason that lower combustion temperature for the algal biochar, making the product more labile. This is the slide showing the methane fluxes relative for the incubated pots. Similarly, like the carbon dioxide graph, this graph showing in the x-axis showing the weeks of incubation and the y-axis showing the methane emission into mu moles per meter square per second. As similarly in the as shown in the carbon dioxide graph, the values of the different biochar types of different doses varied temporarily. Again, the values of the algal biochar among all the biochar types relative to the control value varied from the other biochar types. Overall, the methane fluxes relative to the control values was highest from the algal biochar. This is the table showing the one-way ANOVA for the carbon dioxide efflux and methane efflux to compare over time and the biochar types. The table shows that for carbon dioxide efflux, there was no differences among the incubated pods over time, but the difference was significant for the biochar types for both low dose and the high dose and overall as well. For the methane, a similar pattern was observed where there was no significant differences in the methane emission among the incubated pods over time, but the differences were significant for the biochar type, like for both low dose and the high dose. This was further confirmed by the Tookie's post hoc test analysis, which revealed that the gas fluxes in treatment was mainly significantly different for the algal biochar as compared to the other biochar types. This is a table showing the independent sample t-test for comparing the emission of carbon dioxide efflux and methane efflux for the high dose and the low dose treatments of biochar. This table suggests that there was no significant difference in the emission for both carbon dioxide and methane for any type of the biochar among the between the low dose and the high dose treatments. This is the graph showing the carbon emission in terms of milligram of carbon emitted from the incubated pods. It is showing that the carbon emission in the low dose biochar, which is showing on the y-axis and the x-axis different types of biochar. It shows that the carbon emission for softwood, hardwood, and the bamboo was lower in the low dose as compared to the carbon emission in the softwood, hardwood, and bamboo high dose. On the contrary, the carbon emission for rice husk and algal biochar was higher in the low dose compared to the high dose incubations. Overall, the carbon from algal biochar was remineralized at higher rates than in the other biochar types. This is the table showing the elemental analysis for the selected biochar types, and this showing the highest percentage of carbon for softwood, followed by hardwood, followed by bamboo biochar, then rice husk biochar, and then algal biochar. And for the nitrogen percentage, the amount was highest for algal biochar and the lowest for softwood. This chart overall suggests that the C/N ratio of algal biochar was much closer to the value of the C/N ratio of the algal biochar, which is favorable for the soil microbes. this chart showing the pore water pH for the incubated pots, where the x-axis showing the weeks of incubation and the y-axis showing the pH value for both the low dose incubation pots, like on the above the upper graph showing the low dose values and the lower graph showing the high dose values. Like for both, the pH was alkaline. However, for the algal biochar, the pH was of the algal biochar was lower as compared to the control softwood, hardwood, bamboo, and the rice husk biochar. Similarly, for total acidity, this is the chart showing the total acidity of the pore water on the weeks of incubation on the x-axis, and the total acidity milligram calcium carbonate per liter in the y-axis. It's showing that the highest value of total acidity for the algal biochar for both low dose and the high dose as compared to all the other biochar types. This is the chart showing the salinity of the pore water collected during the incubation period, where the x-axis showing the weeks of incubation and the y-axis showing the salinity in ppt. This is both the low dose, the upper graph, and the high dose, the lower graph. It suggests that the pore water salinity decreased over time. So this could be due to the reason that we watered the incubated pots thrice a week, leading to decreased salinity over time. This is the table showing the ANOVA for the pH, total acidity, and salinity, comparing between the low dose, high dose, and overall. This table suggests that for pH, there was no significant difference among the incubated pods over time. However, the difference was significant in terms of pH over between the biochar types for high dose and for overall. For total acidity, the significant difference was observed mainly for the biochar types, but no difference were observed over time. For salinity, on the other hand, the significant difference was mainly found in terms of time, not for the biochar type. The Tukey's post-hoc test further confirmed that the significant differences in pH and total acidity was mainly because of the algal biochar as compared to the other biochar types. This is the table showing multiple comparisons of salinity during the incubation weeks using the Tukey's post-hoc analysis, as the ANOVA revealed significant differences in pore water salinity over the time. This shows the significant difference was mainly between the week 2 and week 4, then week 2 and week 8, and week 2 and week 12. Suggesting that the salinity decreased over time. This is a table showing the independent sample t-test for pore water pH, total acidity, and salinity. This is suggesting that the pH, the pore water pH for the different biochar type, they did not vary significantly between the high dose and the low dose treatments for each biochar types. The similar trend was observed for total acidity of the pore water of the incubated pods, as well as for salinity, where there was no significant differences between the high dose and the low dose treatment of the different biochar types. So the major findings of the study is that the algal biochar experienced a rapid carbon remineralization and supported high methane emission. The CN ratio of the algal biochar may be more conducive to remineralization because the C/N ratio was more favorable favorable or like more closely matched, which is desired by the soil microbes. The greenhouse gas emissions, they did not vary between treatments like the high dose and the low dose, 10% and 20%, which suggests that higher biochar application rates may sequester more carbon. And for our study, the acid sulfate conditions did not develop, which did not allow us to test the hypothesis that biochar ameliorates low pH conditions.
So I'm just gonna zoom back out and think about like what these findings might mean for management and restoration projects. I think our finding that the more recalcitrant biochars are going to increase carbon sequestration highlights the need for thinking about the type of biochar that you're using in your restoration project. So the more kind of recalcitrant material is probably going to contribute to more longer term carbon sequestration. We expected to find that there would be kind of a big efflux of carbon dioxide at the beginning of the incubation, and that it would tail off and we'd kind of see the transition of, you know, kind of a more of a raw biochar to a biochar that's more recalcitrant and lacks functional groups. But that was not exactly what we saw. It was also interesting that we saw a lot of methane emissions from the biochar that had an algal feedstock. So this, to me, this really highlights the importance of a long-term experiment to understand the stability of biochar in soils. And this has implications much further beyond restoration and wetlands, but is really kind of focused at thinking about whether, you know, the use of biochar in agricultural settings can increase carbon sequestration and help mitigate climate change. It's really important to understand kind of how much of the carbon that's put in biochar sticks around in the soil. Do you want to move to the next slide? Go ahead, Priyanka.
So besides the Wetland Ambassador project that we just discussed, I also had the opportunity to be involved in the restoration projects in the Elkhorn area, as Beth mentioned in the beginning. It's a newly designated Ramsar site, and it's a really beautiful place to work and visit. So the work that I was mainly involved preparing the permanent biochar plots in the Elkhorn area. We also collected the water samples from selected sites of Elkhorn Slough. Besides, we also involved into greenhouse gas efflux measurements in previously installed biochar plots with my colleagues Brittany Wilburn and Lena Chaplin. So overall, the fellowship was really has given me a lifelong opportunity. It was very enriching as it provided me, you know, and like enduring opportunities. So far, I had the experience of working in Indian wetlands, which are mostly the inland wetlands. However, working with Dr. Watson, I got the opportunity to work into 2 different coastal wetlands and experience and understand the differential wetland processes. So the work that I pursued as a part of this research was different from the work that I have been doing. So thus the program really helped me to extend my knowledge horizon and acquiring experiences into different field of research. Working with Dr. Watson really helped me to improve my scientific skill, I must say, by learning new tools and techniques. I really learned new tools and techniques of analyzing the water quality of the wetlands. For example, while working in the Elkhorn Slough, we had to collect the slough water using the ISCO automated water samplers, which I had no idea prior that it can collect the water automatically over the 24 hours by itself. So that is something I have never seen before, or I had no idea. So that is There are many ways that I have experienced different thing, learned different ways of studying different parameters for wetland study apart from the biochar, and also learning new ways of analyzing the wetland water as well. During the study also, I learned how to use the gas flux analyzers as I shown in my slides before. So it was really an overall and a great opportunity for me to improve my understanding on wetland research, as well as train myself into different tools and techniques. However, I find I also got the opportunity to meet to be invited into the SWS 2019 annual meeting, where I could meet the various young scientists and also other wetland scientists, and extend my collaboration, extend my network. So overall, it was a very endearing opportunity and opportunity for me, and I look forward to pursue collaborative research works in future as well, focusing on scientific and sustainable management of wetlands globally. So I would really like to extend my sincere acknowledgement to the Society of Wetland Scientists for providing me all such opportunities and providing me the financial assistance for supporting my research. I am thankful to Drexel University and the Academy of Natural Sciences of Drexel University for providing me with laboratory facilities and financial assistance to support my research. I'm thankful to Brittany Wilburn, one of my lab colleagues at Drexel, for her help throughout the projects. We're still working together, and we hope to work in the future as well. Last but not the least, I am really thankful to my host home institution, Assam University Silchar, for supporting me and allowing me to pursue the research at Drexel. So with this, we come to the conclusion of our talk. So we're happy to receive any questions, and thank you so much to all for listening to us.
Thank you so much to Priyanka and Beth. This was a really great talk, and we already have a bunch of questions in our question pane. So again, just a reminder for all of you listening, I'm going to be reading your questions that you're typing there. And you can upvote certain questions. I'll read those first. If you do forget to ask a question or have to leave before we finish, you can email the presenter at the email address listed. And let's just get started. So it looks like the question at the top is, can you describe the type of algae used to make the algal version of the biochar and the process used to make it compared to the wood-based biochar?
Can I go ahead?
Yes.
Okay, so the biochar that we used for our study was mainly purchased online. So while we inquired about the algae biochar making process, so we got to know that it is a freshwater algae, and the temperature was very low as compared to the other conventional biochar making process. So this is the information that we have from the producers.
Okay, thank you, Priyanka. The next question that we have is, can you estimate how you think eucalyptus biochar would react?
Okay, so I think I'll answer this question, which is basically, I don't have an answer. I don't think that we have a good understanding of what might be unique about eucalyptus biochar versus other biochars. It does seem like it doesn't have as high of a pH as some other biochars, but what that means in terms of soil conditions, I would say that we're still at the very beginning of trying to figure out.
Thank you for that answer. We have another question here. Do you think we can use this biochar not just for restoration, but also for compensation programs, or for example, to create new wetlands?
I can open this up to Priyanka if you want to respond. If not, I can.
Okay, so by compensation, I'm not sure what the hectare is meaning, but I think the role of biochar is also being encouraged for crop productivity, and but again, I don't know, like if you can really switch this fertilizer purpose of biochar by manure. I don't I'm not sure what that does he mean by the compensation program. So Beth, if you would like to take the questions as well.
I think maybe what this means is, can biochar be used for wetland creation that's compensation for wetlands that are destroyed? And so, yeah, so I don't think that there's any reason why biochar couldn't be used in such a project. And there potentially on the state and the regulatory situation, there could be credits of some sort that are able to be utilized to subsidize that cost.
Okay, thank you. The next question is, why is algae biochar so unique compared to the others?
Okay, I can go ahead with this. So as I mentioned before, the algae biochar, it really had a unique pattern as compared to the other biochar type. It's mainly because of the fact that the biochar was produced as a very low combustible temperature, which has led to had led to the labile nature of the biochar, and with the period of incubation, the organic acids from the biochar kind of released at the initial days of incubation as compared to the end days. So overall, the unique result that we found from biochar was mainly because of the low combustible temperature of biochar, making it more labile as compared to the other biochar type. Beth, would you like to add something?
No, I'm looking forward to the next question. I'll take that one.
All right. Well, the next question is, if I understand correctly, the algal biochar would result in conditions most suitable for soil microbes and presumably nutrient availability for revegetation in a restoration setting. Do you anticipate that over the long term, the carbon sequestration through increased biomass would more than compensate for the short-term increase in emissions?
So I think that in the context of coastal wetlands, most of the carbon that is sequestered is actually in the soil. And so this includes the below ground biomass as well as allochthonous material that's trapped by the sediments and remains in the soil sequestered because of the anoxic conditions. And so there's really a lot of different ways that biochar can interact here. So first of all, some studies have found that biochar actually has, because it creates microhabitats that are conducive for microbes, it actually can lead to more turnover of below ground carbon. And so on the one hand, that might increase biomass of the plants, but it might actually decrease long-term carbon sequestration in the soil, which is really counterintuitive. Right, so the question is really asking if the fact that the biochar is turning over, those nutrients are then available to support plant growth. I mean, I think it's a trade-off, but a lot of times in coastal wetland soils, we're really looking at the carbon that's sequestered below ground, much more so than the above ground biomass. this wouldn't be true in other types of wetlands, forested wetlands, mangroves, that sort of thing, but it's true in coastal marshes.
Thank you. This question is a bit of a switch about what we would usually answer. It is, are there any things that Beth and Drexel learned about wetlands in India that Priyanka works on?
So I will say that I don't think that I actually learned too much about wetlands in India, but I did have a lot of conversations with Priyanka and continuing about kind of culture in academia in India compared to the United States. And that's something that I feel like has been a really kind of productive conversation between each other. We've really kind of learned things about what works and what doesn't work, you know, among the two different academic systems. And another thing is that we have a lot of, Priyanka is ethnically Bengali, and we have a lot of immigrants from Bangladesh in Philadelphia, where I live. And I've worked with a couple of students whose families were from Bangladesh and who are very, as you can imagine, very interested and motivated to learn about wetlands and climate change. And so it was really motivating to me to learn, first of all, where Priyanka is from is the very eastern side of India. So India has a very interesting shape, and kind of the long-term reasons for that, and thinking about like the partition and what happened in, you know, India in the last century, I think has really kind of built my cultural competence in working with students whose families immigrated from this part of the world.
Great, thank you for that answer. We probably have time for maybe 2 more questions, so I'll just do the ones that are on the top here. Can we do such a study in an inland saline wetland?
Yeah, I do have a plan or interest of doing such kind of study in Indian wetlands as well. I hope we would be able to do it. Yeah, and the work that is mostly being done in the US is mostly on the wetland restoration project. So I'm really looking forward to such kind of work to pursue in India as well.
And this next question is actually a bit of a follow-up to that, is will you give support if Vipin Solanki will start a biochar project in India? And that's for Priyanka. Will you give support for such a project?
Yeah, I would love to, because I mentioned just now that I am really interested to pursue such kind of work in India as well. And if there is someone who is interested and we can work collaboratively, what else can I ask for? So I'm really happy to be there with all my capacities that I can.
We do have a note in the chat from Rajashri just asking maybe some clarification about it being an inland saline wetland. To if this would work in that condition.
I see. Okay.
I think what they said was that it could work in that, and that's something that they would be, Priyanka was interested in looking into, if I understood her answer properly. Okay. Oh, let's see. Yes, maybe you just want to reiterate, Priyanka, what you had said.
Yeah. Like I really am looking forward to pursue such kind of study in Indian wetlands as well. So far, the study that I have been doing in India, it's mainly focusing on the inland wetlands. I don't have the experience of working any saline Indian wetlands so far, but I'm really looking forward to perform such kind of studies. And with, of course, as Vipin Solanki, if I'm not mistaken the name, he asked me that whether I will be I will support him if he would like to pursue such studies in India. So I am really looking forward to such kind of studies to be done in India as well, from my experience that what I have got by working with Dr. Elizabeth Watson. Thank you.
Great. All right, thank you for answering those questions. We have just a couple of quick but important announcements before we conclude. So the first announcement is that our next free webinar will be held on September 17th, August 20th here, and that's on restoring native clam communities. And this is going to be delivered by Todd Osborne. And our next Spanish webinar will be in September on Mexican mangroves, presented by Dr. Maribel Dominguez. So as a reminder, all of the Spanish webinars are offered free of charge to members and non-members alike. And finally, as we like to say, please be sure to subscribe or follow SWS social media channels such as our Facebook, our LinkedIn, and this will help keep you informed as well as help support the society. We also do have a YouTube channel, which is becoming a lot more popular. That's where all of our webinars are posted about 3 months after their original broadcast. They do have multilingual subtitles. And newly added are also our wetland interviews, one in English and one in Spanish, posted to our YouTube page. You can quickly link, you basically can go to those in the resources tab and the SWS website and select wetland interviews if you want to see those there. And those are some really great resources. We've had some good interviews. If you are Spanish speaking, be certain to subscribe to the Latin American and Caribbean regions Facebook page at the link listed here as well. Oops. And then finally, thank you again to Priyanka and to Beth for their presentation, to our webinar sponsors, and thank you to our audience for participating today. I know I got a lot out of this presentation. I really thank Beth and Priyanka for your time, all of your hard work, and I can't just wait to see what more you guys do. Have a wonderful day, everybody. Stay well.